I will introduce our next presenter, who I know he was on earlier, so there he is, David Pointu, who is a lead application specialist from Cytiva, and am I pronounced Cytiva? No, yes, Cytiva. David. Cytiva. Yeah, that's right. I thought it's. I have it misspelled here. I thought it sounded wrong. Cytiva. Thank you, David. I'm not going to, without much delay, because we have a quick presentation from you to get more insight on multiplexed imaging.
Yeah, so good morning, anyone. I will start to share my screen. So I'm an application scientist working for Cytiva for the imaging portfolio, microscopy imaging portfolio of Cytiva, and then the multiplex imaging system that we just released. I'm very happy to have this communication with all of you for this virtual conference. So I have 5 minutes to introduce this technology to you. So it's a multiplexed imaging solution for the FFP tissue. That will help actually to provide tools for more precise diagnosis. It's really in the cancer research. It can also help the people to inform on what we call the patient stratification, to be exposed to different and the most relevant treatment. It's a tool that can also guide the therapy and the doses, thanks to the information that you will be able to collect. In the aim actually to identify targets for drug development. So I will start with an example. So you can see on the right, it's hematoxylin eosine coloration. Actually, it's a virtual one. It's based on fluorescence, and it's a tool that still can be used, but it's more to say, is it cancer or not? And what the multiplex imaging system can bring to that. If you see this picture, we'll be able to identify what cell types are actually present in the tissue, in the tumor microenvironment. You will be able to detect where are the biomarkers were expressed. And regarding the tumor microenvironment, you will be able to see if it's vascularized. You will see if the immune cell managed to infiltrate the tumor or not. And also regarding the immune cell types, precisely, we will be able to see if they are in or out or around the tumor. And it's also thanks to the fact that we have access to more and more biomarkers, so you can identify the human immune cell lineage, the tumor cells, and more the functional status of the immune system. So quickly, this system, the CellDive system, is actually composed of 3 different parts. The first one will be more related to the biology and the workflow. So first, we have validated a large list of antibodies that you can start and you can use to work with your assay. Then we have developed a specific workflow. I will describe that just a little bit later. A specific acquisition software to guide any user to perform that. Then we have the imager itself, which is based on the fluorescence imaging microscopy, which is also the core of our expertise. And last part, very important, it's the analysis software. And for that, we partnership with Indica Labs, and we are offering the software Hello. If you see now the more detailed workflow, so you can see that we can work with a different kind of tissue, the whole tissue, just a few or large number of regions of interest of the tissue or the tissue microarray. Then you will start the cycling to do the multiplex imaging solution. So we always start with an autofluorescence imaging. It's very important to be able to characterize the natural autofluorescence on the tissue before to start the staining. Then you will perform the staining. So that means that most of the time that will be primary antibody conjugated with with a dye. Then we will perform the imaging with the system. We can do 5 wavelengths at a time, so it's a way also to scale up the experiment. In the background, a lot of image processing and alignment will occur, which is also mandatory because there is different cycles, and this part, which is related to the microscopy, is very important. And then we will inactivate the dye, and then we will be ready to start a new cycle with a new cycle of staining. At the end of all these cycles, you will get the images, and you will be ready to start the analysis to perform the different segmentation, the compartment segmentation. You will make a feature extraction, and the software will classify the different cells, and you will be able to get a deep understanding of the tissue and of the immune response. One very important part is the antibodies. So we have spent a lot of time actually to validate antibodies. So we are close to 400 antibodies that you can start with and ready to be usable. And on the right, you can see that the classification of the different antibodies to see in which categories that you want to start and you want to work with that. So for the conclusion, it was very quick. Huh? So CellDive is very customizable thanks to the fact that you can use a lot of validated antibodies. We give precision, and it's thanks to the microscopy and all the workflow that we have developed. And very interesting, the commercialization is new, but it's a proven technique because it has been developed since almost 10 years, so mainly in the US, but it's the reason that we already have partners, users, and customers, and there is a lot of articles that describe results obtained with the CellDive system.